Hello students, now we will start with new topic, that is molecular basis of inheritance. Here we will read about the molecules that are responsible for inheritance. We know that inheritance meaning is transfer of genetic information or characters from parents to offsprings. And the molecules that are responsible for such inheritance are nothing but nucleic acids. There are two types of nucleic acids existed in living organisms. One is DNA and other is RNA. DNA is a nucleic acid and it is defined as a polymer of nucleotides. RNA also the same means nucleic acids are considered as polymers of nucleotides. Polymers means many units of nucleotides. What are these nucleotides? Nucleotides are composed of pentose sugar. In a sense, the compound that is having 5 carbons. Next is phosphate molecule. Next is nitrogen bases. These nitrogen bases are basically of 5 types when we combine both DNA and RNA molecules. Nitrogen bases in total are 5. What are those? Adenine, guanine, cytosine, thymine, and uracil. Adenine and guanine are called as purines. Adenine and guanine are made up of 2 rings, so they are also called as dicyclic compounds made of 2 rings. So they are also called as dicyclic rings or dicyclic ring forms. What are those 2 rings? One is benzene ring, other one is imidazole ring. Next, these 3 nitrogen bases are called as pyrimidines. These are made up of only one type of ring, that is benzene ring. Only benzene ring. They are also called as monocyclic ring forms or monocyclic compounds. We will see some more points regarding DNA. DNA was first discovered by Frederick Mister in 1869, and term DNA was given by Zacharis in 1881. Frederick Mister has found this DNA in nucleus, and DNA also exists in nucleus of pus cells. Next, DNA also exists in mitochondria of both plants and animals, and also in chloroplast of plants. The DNA in the nucleus of pus cells, it can be considered as double-stranded structure. In case of mitochondria and chloroplast, the DNA is double-stranded circular DNA. The DNA is also double-stranded and circular in prokaryotes also. Double-stranded I am writing with small d and small s. Double-stranded, circular and naked DNA. Why we are using this term? Because in case of prokaryotes, the nuclear membranes are absent. No nuclear membranes is seen in prokaryotes, and DNA is dispersed in the cytoplasm. In case of prokaryotes, the DNA is embedded along with some gelatinous material. So, the total structure of gelatinous material plus double-stranded circular naked DNA together are called as nucleoid. So, DNA plus gelatinous material together called as nucleoid. Now, DNA, the term is given by, already I have given, I think so. Now, DNA also exists in double-stranded helical structure in eukaryotic organism. That structure was explained by Watson and Crick. The double-stranded helical structure of DNA was explained by or was given by Watson and Crick. Based on X-ray diffraction method given by Wilkins and Franklin, and also based on Erwin Chargaff's rule. X-ray diffraction method and Erwin Chargaff's rule. What is this method of X-ray diffraction method? By exposing the X-rays onto a substance, the atomic and the molecular structure, nothing but the crystallographic structure of a material can be known. Means, by exposing a material to X-rays, the atomic and molecular structure this we simply call as crystallographic structure. Can be known. It is applicable only to DNA. According to him, the number of purines is equal to number of pyrimidines. This we can represent as A plus G, which are nothing but adenine, guanine together, means the sum of purines is equal to sum of pyrimidines, C plus T. And it is not only should be equal, should be same, it must be equal to 1 and should be equal to 1 or equal to 1. This we can again write like A plus G divided by C plus T, means adenine plus guanine divided by cytosine plus thymine. The answer should be equal to 1. Another point, the base ratio, means nitrogen base ratio, if adenine and thymine are greater than the content of guanine and cytosine, such type of DNA is existed only in eukaryotic cells. If base ratio of is greater than guanine and cytosine, then such DNA is called as AT type of DNA. And the ratio is more than 1. Base ratio will be more than 1. Such type of DNA, means AT type of DNA, is seen in eukaryotic cells. Only in eukaryotes. Another point, if the guanine content or guanine and cytosine or G plus C is greater than AT or A plus T, then such a type of DNA is called as GC type of DNA and it is seen in prokaryotes only. Prokaryotes. So, base ratio should be constant. So, base ratio, how can we represent base ratio in the sense A plus T, G plus C? We can write like this, is always constant for a given species. Means it varies from species to species. Melting point of DNA, it depends upon the amount of guanine and cytosine. Guanine, cytosine amount or quantity. Next, the melting point is more in case of prokaryotes. So, in prokaryotes, due to more guanine and cytosine bases, the melting point is more. Next, DNA absorbs UV light or UV rays at 2600 angstroms of wavelength. This we represent with lambda. DNA as a polymer of nucleotides has pentose sugar, that is deoxyribose sugar. Next, nitrogen bases. Nitrogen bases as adenine, guanine, cytosine, and thymine, and one phosphate molecule. If you see the biochemical structure of this adenine, guanine, cytosine, and thymine, how the structures are going to exist. First one, adenine. Adenine is a purine. It is made up of two rings, so called as dicyclic compound. Two rings. What are those two rings? One is benzene ring. Another is imidazole ring. So this is called as dicyclic compound. The biochemical structure of adenine is if you give the numbering 1, 2, 3, 4, 5, 6, 7, 8 and 9. At 6th carbon position there is amino group. At 2nd carbon position there is one ketone group. Sorry, ketone group is not present. At 3rd carbon position there is nitrogen atom, and 1st carbon position also another nitrogen atom. 7th carbon position also other nitrogen, and at 9th carbon position. If this is benzene and imidazole ring, the ratio of nitrogen atoms is 1:1 or 2:2. So in the benzene ring 2 and imidazole ring 2. So adenines are considered to be having nitrogen atoms at 1, 3, 7, 9 positions. Next, about guanine. Guanine is also a purine. It is also made up of two rings, that is one benzene ring and another imidazole ring. It is also called as dicyclic compound. Now, the biochemical structure of guanine with one benzene ring, another imidazole ring, will give numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. At 1st and 3rd carbon position there are nitrogen atoms. At 7 and 9th positions also nitrogen atoms, benzene ring, imidazole ring. At 6th carbon position there is amino group, one ketone group, and at 2nd carbon position there is amino group. Means at 6th carbon position of adenine, in case of guanine, it was shifted to 2nd carbon position. Okay. Cytosine. It's okay. We'll write again cytosine here. Cytosine of DNA, it is made up of only one ring, that is benzene ring. Next, cytosine is a pyrimidine. So it is also called as monocyclic compound. It is also having nitrogen atoms, but at 1 and 3rd positions of ring. If we go for the structure of cytosine, only one ring, that is benzene ring. If you give the numbering 1, 2, 3, 4, 5 and 6. Now, here we should represent the nitrogen atoms at 1st and 3rd carbon position. At 4th carbon position there is amino group. At 2nd carbon position there is ketone group. Okay. Next, about thymine. Thymine is nitrogen base and the pyrimidine that is present only in DNA. So it is also a pyrimidine, monocyclic, and it is made up of only one ring. So monocyclic, that is benzene ring. If we go for the structure, it is having ketone group at 4th carbon position and 2 nitrogen atoms at 1 and 3rd carbon positions, another ketone group at 2nd carbon position, and 1 methane group at 5th carbon position. Thymine is also called as 5-methylated uracil, because uracil compound in case of RNA is not having this methane group, then it will be uracil. But the same uracil, if it has attached 1 methane group at 5th carbon position, then it will be converted into another nitrogen base, that is thymine. So we also call thymine as 5-methylated uracil. Purines bind to pentose sugar in a nucleotide at 9th nitrogen atom position of purine and or to 1st carbon position of pentose sugar, that is deoxyribose sugar. If you see the structure, if this is the pentose sugar, 1, 2, 3, 4 and 5th carbon positions of a pentose sugar, if are like this, then this is imidazole ring, this is benzene ring, carbon numbering 1, 2, 3, 4, 5, 6, 7, 8 and 9. This 9th nitrogen atom position is binded to the 1st carbon position of the pentose sugar, and the bond formed here is called as glycosidic bond. You will see the complete big structure of this when I explain you about nucleotide biochemical structure. Next, coming to pyrimidines. Where are these pyrimidines binded? Onto the pentose sugar. Pyrimidines bind to pentose sugar in nucleotide at 1st carbon position, or we'll write about first pyrimidine, 1st nitrogen atom position of pyrimidine to 1st carbon position of pentose sugar. Means, if we draw the pentose sugar structure like this, carbon 1, 2, 3, 4 and 5 of pentose sugar, the pyrimidines are attached at 1st nitrogen atom positions. So here will be one bond, 1, 2, 3, 4, 5, 6. You can take any of the pyrimidine here, cytosine, thymine, etc. Even in case of RNA also, if it is uracil, then still it will be binded from its 1st nitrogen atom to the 1st carbon position of pentose sugar. Again, we call this bond is as glycosidic bond. What is the strength of this glycosidic bond? We will equalize this one to the strength of covalent bond that you study in chemistry. So this is about the nucleotides and the nitrogen bases, their biochemical structures, and the positions where these nitrogen bases are binded to pentose sugars of the DNA, that is deoxyribose pentose sugar. Now we will see the structure of pentose sugar, because till now we have seen the biochemical structures of nitrogen bases. Now we will see about pentose sugars. Nucleic acids are of two types, that is DNA and RNA. Okay, now we are discussing basically about DNA and its nucleotide and their biochemical structures. Just we will have one glance regarding this RNA pentose sugar also. The pentose sugar, because we should know how the ribose sugar of DNA was formed. is called ribose. The structure of a ribose is like this, means the ring form. We will give the carbon numbering as carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. Carbon 1 is having hydroxyl groups. Carbon 2 also is having one hydroxyl group. Carbon 3 is also having hydroxyl group. Carbon 4 is also having hydroxyl group. And carbon 5, it is replaced with CH2OH, that is alcohol group. Now, the oxygen at second carbon position, the oxygen of the hydroxyl group at second carbon position, it is binding with all types of metallic and non-metallic compounds, whatever that has come nearby it. For suppose, RNA is considered to be the first existed genetic material. And inside the nucleus of eukaryotes, there are metals like zinc, etc. So easily it is used to bind with that of metal. Whatever the metal that has come nearby, this oxygen is going and binding to that, so that there is some changes or variations genetically seen in case of living organisms. So that in course of evolution, there are drastic changes among and within the living organisms. Any type of living organism basically tries to survive in the nature. So in course of that survival, RNA has removed this oxygen because of oxygen, the total RNA has attained an instability or unstable nature. Because this oxygen only at second carbon position, it is easily binding with whatever the metal compound that has come nearby it. So that it has removed that oxygen at second carbon position and it changed its structure into carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5, which is occupied with CH2OH. At the second carbon position, it has removed that oxygen, so only hydrogen existed in the next structure. Other distributions of hydroxyl groups is the same. Nothing change is seen in any of the carbon number of the changed form of pentose sugar. The pentose sugar of RNA is called as ribose, but it has removed its oxygen atom from second carbon position. So, because of that removal, the name of the ribose sugar was changed to deoxy, means oxygen was removed at the second carbon position, deoxyribose sugar. Now, because of this deoxyribose sugar, the total genetic material has attained stability and also a repair mechanism was developed in complete genetic material. Deoxyribose sugar is the pentose sugar, means 5 carbon compounds that existed in DNA. Now, the RNA, because of the change, one of the change in pentose sugar at second carbon position, it has been converted into DNA. One more change is that based on the nitrogen basis also. Now, DNA, due to stable nature, has attained a special feature called repair mechanism. Next, and DNA can exhibit autocatalysis. Autocatalysis. What is meant by autocatalysis? Means DNA fragment giving out rise to new DNA fragment is called as autocatalysis. We also call as DNA replication method. DNA giving out new DNA, forming new DNA. So we also use this term to explain DNA replication. Phosphate molecule, it exists with one double bond oxygen and three hydroxyl groups. And phosphate molecule binds one of its hydroxyl group to the third carbon position of the pentose sugar and other hydroxyl group to the fifth carbon position of the pentose sugar with help of ester bonds. So we will see the biochemical structure of a nucleotide. The deoxyribose pentose sugar, carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. At second carbon there is hydrogen and hydrogen. At first carbon there is OH and H group. At third carbon there is OH and H group. At fourth carbon also one OH group, and at fifth carbon position there is CH2OH. Now a phosphate will be binded because we know that nucleotide is a combination of nitrogen base plus pentose sugar plus phosphate molecule. Now we have represented pentose sugar, that is deoxyribose sugar, which is composed of 5 carbons. Now at the fifth carbon position, if a phosphate has to be existed here, there should be 3 OH groups. Now the hydrogen of pentose sugar will be binded with hydroxyl group of the hydrogen of the phosphate molecule will be binded with OH group of the pentose sugar, and it is removed in the form of water molecule. Now, there will be a bond that is formed between oxygen of phosphate molecule and the carbon. We call this bond as ester bond. Now, what about the nitrogen base attachment to the pentose sugar? At first carbon position, if it is a purine or pyrimidine, we will take, is having only one ring. If we take cytosine, then we will give the numbering carbon 1, 2, 3, 4, 5 and 6. At fourth carbon position there is amino group. At first carbon position there is nitrogen atom, and third position also nitrogen atom. Okay. Now this nitrogen is attached to the first carbon position of the pentose sugar. Already we have seen that name of the bond that is formed is glycosidic bond. So a nucleotide which is composed of nitrogen base, pentose sugar, phosphate molecule will be like this, with one ester bond by dehydration and glycosidic bond. Here also you will see the loss of one water molecule, because already in the previous structure we have seen existence of OH and hydrogen atoms, compounds there. So this is the structure of a nucleotide of DNA. Nucleotide, will write that also, of DNA. In case of RNA, here the nitrogen base changes, but everything structure will be similar. Existence of RNA in cytoplasm was first proposed by Alexander Rich in 1962, and the term RNA was given by Gilbert in 1986. Term RNA was given by Walter Gilbert in 1986. RNA also exists in an enzyme form called as RNase and ribosome. Means the RNA that is behaving as enzyme also, the sentence is same. We'll write that only. RNA that behaves as enzyme is called as RNase and ribozymes. Next. RNA is also a polymer of nucleotides. But the nucleotides in case of RNA are composed of pentose sugar, that is ribose, plus phosphate molecule, that is phosphate molecule, and nitrogen base, what are nitrogen bases in case of RNA? Here also we will see purines and pyrimidines. Purines are adenine and guanine, same like that of DNA. Pyrimidines are different from that of DNA pyrimidines. The cytosine is the same, but instead of thymine, there is uracil. The biochemical structure of uracil is, uracil is also a monocyclic compound, means it is made up of benzene ring. The structure looks like since it is a pyrimidine, it will be having nitrogen atoms at first carbon position and third carbon position, and at fourth carbon uracil is having one ketone group, and at second position another ketone group. So this is a benzene ring. We have seen thymine is also having one ketone group at second carbon position, another ketone group at a fourth carbon position, but extra methane group at a fifth carbon position. This one we have seen in case of thymine in DNA. So thymine is also given a term like 5-methylated uracil, because if you see the structure, uracil and thymine, the structures are same except the methane group at fifth carbon position in thymine. Apart from that, everything structure is same. 2 nitrogen atoms at first and third carbon position, 2 ketone groups at second and fourth carbon positions. So thymine is also called as 5-methylated uracil. Nucleotides. Names basing on type of nitrogen base. If the nitrogen base is adenine, then the term of the nucleotide is adenylate acid. If the nitrogen base is guanine, then the term is guanylic acid. If the nitrogen base is thymine, then the term of nucleotide is thymidilic acid. If the nitrogen base is cytosine, then the term is cytidilic acid. If the nitrogen base is uracil in case of RNA, then again the nucleotide term is uridylic acid. This is about nucleotides, other terms. If we know that nucleotide is a combination of nitrogen base plus pentose sugar plus a phosphate molecule. If a phosphate molecule is removed, then the same nucleotide is also called as nucleoside. Nucleoside is another different word that is given for a combination of pentose sugar plus nitrogen base. We can also call the same nucleoside as nucleotide minus, this is minus phosphate. So here again the terms differ. First one, if the nitrogen base is adenine, then the term is adenosine. If the nitrogen base is guanine, then term for nucleoside is guanosine. If the nitrogen base is cytosine, then the term is cytidine. If the nitrogen base is thymine, then nucleoside is thymidine. If nitrogen base is uracil, then nucleoside is uridine. We know that thymine exists only in DNA, uracil exists in RNA. So this is about the introduction part of molecules that are majorly responsible for inheritance or transfer of genetic material or the characters from parents to offsprings. We will see few other points, that is nucleic acids, because we have finished about both DNA and RNA. Now we will see one or two points regarding the nucleic acids. Nucleic acids term is given by Altman in the year 1889. Altman also called nucleic acids as nuclein. Nucleic acids as nuclein. Next, DNA, the genetic material or the nucleic acid, a polymer of nucleotides, is considered as acidic in nature.